Thank you. Let us start by imagining a sensor which is so small that it can go inside the cell like a Trojan horse. And once it is inside the cell, what can I do? I can basically spy on my cell to tell me the real-time free radical level inside it. So if that's what interests you, I'm Harsh Jain. I'm a PhD student in Romana Shirhagar's group. And what I do is use nanodiamonds as quantum biosensors to study the real-time free radical levels inside CHO cells. And the hamster that you see on the screen, it's not just for motivational purposes, even though it does motivate me, but it has something more. So we'll see that. First of all, let me introduce you to the idea of fluorescent nanodiamonds and how they are different from traditional nanodiamonds that we are used to. So in a traditional nanodiamond, you can see a carbon lattice, but in this case, if you take out two carbon atoms and basically substitute with a nitrogen and a vacancy next to it, what you achieve is a so-called NV center defect. And this defect, unlike all other defects, actually makes the system more useful. And how does it do that? It makes the system optically probable, so I can basically use visible wavelength of light to probe it, and this system is now very sensitive to magnetic noise around it. So what is our sensing principle? Essentially what we do is we shine the laser on the nanodiamond, and then we study how bright it is and how long does it stay that bright for. And that can give us an idea about the magnetic noise, because this process, where it goes from the bright state to the dim state, this transition time is very sensitive to the magnetic noise. So essentially what we do is T1 relaxometry. We try to plot the curve of this T1 relaxation time in presence of noise, and we see that the relaxation goes down. So the key takeaway from this sensing principle is that higher the magnetic noise, the lower your T1 relaxation time will be. Now, how does this relate to cells? If I take my nanodiamond inside the cell, what am I sensing? What is the source of this magnetic noise? It's free radicals. And what are these free radicals? As Travis also mentioned in his talk yesterday and many more after him, that these are basically reactive molecules which try to scavenge electrons around in the cell. And that's why it's so detrimental that all of the disease models like cancer, neurodegenerative diseases, you see that the markers of ROS are very important because cells under stress always generate these reactive oxygen species, which possess this unpaired electron, which can provide that magnetic noise around my NV center. And that is the magnetic noise that I probe inside the cell. So we take the nanodiamond inside the cell, and then we plot the same T1 relaxation, and we see how much the time goes down in presence of any magnetic noise, which comes from the ROS. And then we have basically a label to quantify how much ROS is being produced inside the cell. And what benefit does this give us over the traditional dyes that we already have? First of all, it's at single cell level. So if I have a population of many cells, I can essentially study a single cell and then check the radical load inside it. It also gives me nanoscale resolution because I can take my nanodiamond inside the cell at a particular location targeted to mitochondria. And then it is doing everything real time while keeping the cell alive. So with this, now comes into picture our hamster. So basically, why the CHO cells? These CHO cells are Chinese hamster ovary cells. And the best part about them is they're already having a decade of literature and well-established SOPs, and they're extensively used in biopharmaceutical industries. Preparation of around 70% FDA-approved pharmaceutical drugs is basically being done in bioreactor tanks, where these cells are being cultured extensively. And that makes them an opportunistic model to study the ROS levels. They have a very high transfection yield, so you can use these cells for protein production assays and reporter assays, and they already have a clinical relevance. So before sensing, first of all, we have to establish that this model actually works for us. And how do we validate that? We have to confirm that our FNDs, fluorescent nanodiamonds, are actually going inside the cell, and I'm not measuring anything outside. How do I do that? It's a very simple procedure where you grow the cells in a flask, in a growth medium, and then in this growth medium, you add some of the suspension of your nanodiamonds. The cells, by the nature of endocytosis, basically uptake these diamonds by themselves. What I'm trying to determine with this cell model is how much time does it take for the diamonds to go inside, and what concentration of these nanodiamonds should I use for them to uptake it efficiently. Once it is inside, you can ask me questions about this entity is actually foreign to the cell. You do not usually find nanodiamonds inside cells. So does it actually harm the metabolism of the cell? So that we validate through confocal fluorescence imaging that the diamond is inside. And we also check cellular viability with plate reader assays that I'll talk about soon. And then once the model is validated, we can take it forward towards quantum sensing and then checking the same free radical load under stress of these cells. So towards the confocal fluorescing data, you can observe as compared to the control, if I keep my time incubation constant, I grow these cells for 24 hours in the presence of diamond. As compared to the control, you see that at 1 microgram per ml, you hardly see any diamonds inside the cells. But at 10 microgram per ml, you see that it's a possibility that cells can even have more than 1 nanodiamond. Now we basically talk about the concentration dependent effect. But if I take it forward to study time incubation, if I stick to 10 microgram per ml but study different time points, then I observe that it is efficient only at 24 hours, which tells me a little bit more about the internalization mechanism of these cells. So now that we have given insights on the time and the concentration, the question remains, does the cell actually get harmed by these nanodiamonds? So that we have to confirm using a cell viability assay. It's an ATP-based luciferase assay. How it works is basically this. You have a cell population. These cells are growing ATP as their metabolite. And then you check after you incubate the diamonds, does the ATP generation get affected? Are these cells still metabolically active? So this we see in our result that as compared to the control where no FNDs were introduced, even the cells which have different nanodiamonds incubation, they do not show any harm in their metabolism. This confirms that fluorescent nanodiamonds do not influence the cellular metabolism. But now comes the question, the story does not end here. Once the diamond is inside to basically sense ROS, but if it's actually sensing the same quantity that it induces, then my sensor is not that efficient. What I mean is, if I take this sensor inside the cell, and if it's generating ROS by itself, then I cannot use it as an efficient sensor. So this we confirm with the DCFD ROS-based assay, where we have a dye which can tell us about the ROS levels. And then we see as compared to the controls where I did not add any diamonds, again, there are no significant differences where the diamond was introduced. And as compared to the positive control where hydrogen peroxide is used to induce that stress, we see that it is highly significant different. This confirms that fluorescent nanodiamonds preserve the baseline radical load inside the cell. So then my sensor does not increase the ROS levels by itself. This leads to our conclusions that first of all, these CHO cells, they're really happy in uptaking the diamonds. So they confirm the internalization with concentration and time-based effects. And then the biocompatibility is also preserved because the diamonds by themselves are not harming the cells at all. And then the ROS is also not perturbed by the system. So we basically use these sensor and they're inert inside the cell. So whatever dynamics that I'm reporting, they're irrespective of the sensor. So this basically leads me to draw this conclusion that I have a biocompatible cell internalized sensor that is ready to report free radical dynamics in living CHO cells. I would like to acknowledge the support that I've received from Yomic facility, which is the imaging facility in our department, and also my group.